Hello and welcome to the third and final lecture on MR spectroscopy for Enigma-U. My name is Ivan Kirov. It's been a real pleasure making these slides and presenting them. Today the topic is data processing. Let's start the presentation. And we'll start with a comparison of conventional MRI and MRS in terms of how the results are interpreted. For conventional MRI, a radiologist looks at the contrast which is generated between gray matter, white matter, and CSF. The type of contrast is determined by the type of image the radiologist is looking at. The image could be T1 weighted, T2 weighted image, and so forth. And the radiologist knows what to expect, what kind of contrast between gray matter, white matter, and CSF to expect for each of these sequences, and he can identify abnormal contrast, can identify whether an image anatomy looks normal. On the other hand, with MRS, we need to end up with a number at the end of the processing. A number which we can then compare to a normative database to assess whether that number represents normal or abnormal metabolism. All right. So here are the key steps, the key processing steps for MRS data. As a whole, all the processing steps are known as a processing pipeline. Well, let's go over each of these. The first step may be to improve the quality of the raw data. Here are common causes for imperfections, which we can deal with in one way or another. Spectroscopic data tends to be noisy, and filters can be used to decrease noise. This process is called apodization. There may be phasing errors. We need to make sure that the spectra are upright and symmetric. Here you can see in the example the difference before phasing and after phasing. There may be data from multiple receiver channels. You can see here on the snapshot from the scanner, the arrows are pointing to whitish objects on the left-hand side of the brain. These represent the receiver channels. All the data gets received by each of these channels, and it needs to be combined into one before continuing. We need to account for subject motion. You can see here in this zoomed version of the NEA peak of the different of the many averages that were required in this experiment that one average is off. That is probably a result of motion, and there is software that we can use to discard this average because it makes the data more noisy. Scalar drift and eddy currents are due to unwanted changes in the main magnetic field, and there is tools to deal with those. And the paper that you see referenced here is a very good resource for this first processing step. Second step commonly is quality control. Quality control is usually automated and visual. It's usually based on established thresholds for SNR and peak line width. These two concepts I introduced in the last lecture as very key, and they are used to assess quality. Additional parameters include the presence or absence of artifacts. I want to discuss this spectrum for a little bit. The low SNR arrow is pointing to the NEA peak, and the NEA peak indeed seems to be, well, it is indeed lower in amplitude compared to the NEA peaks that I've shown previously. And thus, it has lower SNR. But this spectrum would not be excluded based on this. It's lower SNR, but it's actually most likely biologically lower. This spectrum is showing lower NEA. But if the peak height or amplitude was down to the level of the noise, that would be a reason to exclude this data. There's a peak line width. Very wide peaks are undesirable because they impede quantification, and there are certain accepted thresholds as to when a peak is deemed too wide to exclude the spectrum. if there's lipids in the spectrum, so you can see where the lipids may fall in the spectrum, they would be on the right side of the NEA. It may be a cause for concern that these lipids are a result of contamination, because biologically we usually do not see lipids in biological tissue. So if there is lipid in your spectrum, it could be a result of contamination, but it may be okay if they do not overlap with the NEA peak. So that needs to be assessed. If they do overlap with the NEA peak, the spectrum would need to be discarded. Okay, and this would mean that we are ready to determine the areas of each peak. A very important process called fitting. The first step of this process is to run a computer simulation, which will tell us what peaks are expected at what frequencies, given the experimental parameters. So the key experimental parameters we already went over in the second lecture. The TE, or the echo time, the TR, the repetition time, are very central to the software. Software also would need to know the magnet strength and the type of sequence that you're using, and it generates these simulations, which collectively are known as basis sets. You can see each metabolite is expected to have different spectral appearance, with some have single peaks, some have multiple peaks, and the peaks have different appearances based on the metabolites. This is given by the simulation. This collection of individual spectra for individual metabolites is combined, and that gives you the basis set. This basis set is overlaid onto your raw data, and that's how peak areas get estimated. And now it's time to convert those peak areas to results that we can interpret. For example, concentrations. To get concentrations, the signal is calibrated against a reference with known concentrations. A very common way of doing this is with brain water. I told you in the first lecture that water is an enemy in MR spectroscopy, that we try to get rid of it by suppressing it, because its concentration is so big that if you don't suppress it, it interferes. It actually obscures the much smaller metabolite peaks next to it. But in this case, if we're using water referencing, we want the water. So we acquire one spectrum where we do not suppress the water. One spectrum that we want the entire water peak. And we use it as a reference for estimating metabolic concentrations. How? Well, because we know approximately the concentration of water in the brain, in white matter and in gray matter. So we infer what concentration is related to this water peak that we measure in the unsuppressed acquisition. And we scale our metabolite peaks to it, and that's how we know how much of the metabolite there is. The same idea goes when you do external referencing, such as phantom replacement method, shown here. A phantom is a container similar in size to the human head. It is put into the scanner after the subject, and the same experiment as was done in the subject is ran on the phantom. And the idea is the same as with the water, because in the phantom, we know the metabolic concentrations. We actually know them exactly, how much there is of each metabolite in the phantom. So we can scale our experiment to the peak areas that we obtain from the phantom. But there's a lot of correction factors that also we need to take into account to get to concentration. So it may stop at the level of institutional units. We calibrate against the reference, but we don't do all the other corrections that need to be done to obtain concentrations. If we stop at this step, this is perfectly fine. We can compare these institutional units to institutional units acquired from a normative data set. The third way of getting interpretable results is ratios. Results are expressed as metabolite ratios. Most commonly, creatine is chosen as a reference. goes in the denominator. This method takes care of a lot of experimental parameters that you need to account for when you're doing concentrations and even institutional units. These parameters cancel out in the ratio, so the ratio is very convenient. It's widely used because of that. But sometimes it does not allow us to determine if the numerator or the denominator changes, or it gives us somewhat less specificity. A key step in getting into interpretable results is to account for what kind of tissue we have in the voxel, and also do we have CSF in the voxel. Why? Because gray matter and white matter have different metabolic concentrations. For example, there is more NAA in gray matter compared to white matter. So if you have a voxel with varying amounts of gray matter and white matter, you're comparing those concentrations to a voxel with different amounts of gray matter and white matter, the comparison is not fair. The comparison will not be fair also if you have different amounts of CSF between voxels, because CSF contains no metabolites. So if you have two voxels, one has more CSF than the other, but you're getting the same amount of NAA from both, that would mean that the voxel with the more CSF actually has higher concentration of NAA. The tissue inside it has higher NAA concentrations. So you need to account for CSF to get to this conclusion. Okay. By the way, for ratios, you don't need to account for CSF because that's taken into, that cancels out in the ratio. All right, and that's it. The end result of an MRS experiment is concentrations in institutional units or ratios. So the processing is done. Now, the final step is to run a statistical analysis to compare these concentrations, institutional units or ratios to the normative data set. That's how you determine if metabolite levels in the tissue of interest are normal or not. And these slides, this and the next slide you've seen. On this slide, you will see common results that you may get. This is how you would interpret them. We went over this before. These are the main results that you may get: low NAA, high myoinositol, high choline, high creatine, and each has an interpretation linked to it. Of course, there's many other possible results, and this slide is just one possible interpretation of them. Okay, and with these references, I will say thank you for your attention.